And she got her start at the University of Witwatersrand in South Africa. She was both a speech pathologist and an audiologist. Got her bachelor and master's there. Then she came to the US and got her PhD in auditory neurophysiology at LSU in New Orleans. And then through a series of promotions and outstanding research, she is now professor of otolaryngology and molecular and integrative physiology at the University of Michigan and a member of the Engineering Research Center for Wireless Integrated Microsystems. And she's a great example of a neuroscientist who is now applying her findings to develop clinical biomedical engineering devices. And you'll see where that's going. So it's a great mixture of neuroscience and now biomedical engineering. So, Susan.
Thank you, Bob. So if you guys right at the back can't hear me, come forward because I don't have a loud voice. And so if you can't hear me, it's your fault because there's it's right over here next to me. So, and I'm also going to be sitting down, if that's okay with everybody. So if you can't see me, come over here. So the title of this talk is Multisensory Neurons in the Cochlear Nucleus Contribute to Phantom Sound Perception. And so the talk is about tinnitus. It's not really about tinnitus, but it's about mechanisms in the brain that might contribute to how we develop tinnitus. How many people in this room have tinnitus?
Tinnitus is ringing of the ears. Ringing of the ears.
Okay, put it this way. How many people in this room have ever had tinnitus? Okay, so that means it's a fluctuating hearing loss. It goes with hearing loss. It's a fluctuating disorder. And this, the audience here is too young, because if I give a talk to an audience that has a lot of full professors in it, then everyone raises their hand without, saying it's intermittent. So it's usually associated with hearing loss, but it can also occur in the presence of normal hearing thresholds. And it used to be until recently that if you went to the audiology clinic and you got your hearing measured and they said your hearing was normal, you had normal thresholds and it was assumed that you have normal hearing. But some recent studies by a group in Boston, headed by Charlie Lieberman, has shown that if you noise damage animals, this includes mice and guinea pigs, and then you give them the kind of noise damage that produces a temporary threshold shift. So you blast them with noise, then you measure their hearing, their thresholds come back to normal, and that's what you're seeing over here. So this is actually plotting the level of the sound, and this is, you can't really see here, but this is the amplitude of the ABR, auditory brainstem response, the first wave, which represents the auditory nerve. So if you look here that the control is the green. So here we're looking at thresholds over here. And the black is after 8 weeks, and you can see that the thresholds are normal after 8 weeks. But if you look at super threshold levels, so here we're plotting the amplitude of the wave 1, this is actually emission, sorry, come to this one here. You've got the amplitude of wave 1 here. The thresholds are back to normal, but you can see that the amplitude of the wave 1 at higher levels is much reduced, and especially at certain frequencies. And so what this has pointed out to the whole auditory community is that if you go into the clinic and you measure thresholds, that doesn't mean that super threshold hearing is normal. And so I will come back to this later on, but I just wanted to preface that with, so when people say that tinnitus can occur in the presence of normal hearing, it probably isn't normal hearing. It's probably normal thresholds, but there's some dysfunction. And what the same group went on to show was that they looked at the synaptic ribbons in the cochlea and showed that these abnormal rate level functions were associated with a loss of synaptic ribbons. So where you saw those rate level functions where you couldn't get the amplitude up to its normal level, that correlated with a loss of synaptic ribbons right at the hair cell in the cochlea. So it probably is the case that almost all cases of tinnitus involve some form of hearing dysfunction. not threshold loss. And so what the tinnitus world now is sort of looking at is that the general concept now is that tinnitus is maybe initiated by hearing loss, but it's a result of central plasticity changes in the central nervous system as a result of that. Yes, do you have a question?
Just so I'm clear on the topic of tinnitus, since that's going to be the main talk of the focus of the talk. I get ringing of the ears, but I thought tinnitus required an actual emission of sound.
Oh, no, not at all. No, that's a completely different phenomenon.
Which is a different phenomenon? The emission or the ringing of the ears?
They're different from each other.
Okay. Okay, so I want to be clear. So we're
Yeah.
not talking about ringing of the ears. We're talking about where you actually have emission.
No, we're talking about tinnitus.
So tinnitus is the first one. Okay.
Tinnitus is the subjective perception of a sound when there isn't a sound. So it's like phantom limb pain in the sense of when you cut off your arm. I mean, not like you've cut off your arm, but when people have cut off their arm, they can still continue to feel the presence of the arm there, or pain in the arm, even though the arm is not there.
So you're saying my subjective perception of ringing is due to loss of ribbons?
Well, you have to listen to the whole talk.
Okay. I just want to make sure I was clear.
Well, what I'm saying is that it's got something to do with it, and you'll see what it is. And so this is in a way sort of a summary of what it has to do with, but you'll see that the picture is very complex. And so what this is showing, this is a study that was done by a group in London, where they took people who had normal thresholds, and they had tinnitus or they didn't have tinnitus. And so what they showed was this is the normal person. So they had normal hair cell complement, presumably, because their wave 1, which represents firing of the 8th nerve, the auditory nerve, was normal. And wave 5 was normal. So these are like normal. And then in the tinnitus group, what they showed was wave 1 amplitude was smaller, just like I showed in the previous slide. And that's suggesting that these people had some degree of hearing impairment, even if their thresholds were normal. But what you're showing here is that wave 5, which is representing the firing of the inferior colliculus, was back up to normal. And so what that means is that even though there's auditory nerve input to the brain may be reduced, the brain is making up for that with some sort of central gain mechanism that brings this back up. And so that's sort of the concept that I'm going to be alluding to when I show you some of the work from our studies. So something in the brain is causing this response gain. It could be decreased inhibition, or it could be increased excitation, which is what I just said. Okay, so like I've just said, tinnitus is usually associated with hearing loss, but it can also occur after head or neck trauma, whiplash, or dental work, by the way. So there are reports of people who got a tooth abscess, they got tinnitus while the tooth abscess was there, then the tinnitus went away. There's a much higher incidence of tinnitus in people who have TMJ disorders. 50% higher incidence of tinnitus. So there's something here in the trigeminal innervation, because it's mostly head and neck where these disorders contribute to tinnitus. Something in the trigeminal innervation of the brain that has something to do with tinnitus. And so another clue about that is a phenomenon called somatic tinnitus, which really refers to a large group of tinnitus. 2/3 of people who have tinnitus can modulate their tinnitus by clenching their jaw, or thrusting the jaw forward, or pressing on their cheeks or their forehead or something, tensing their neck. And this has been investigated by quite a lot of people. So different kinds of manipulations can change the tinnitus. It can get louder, it can get softer, its frequency can change. So the trigeminal system innervates jaw muscles, joints, tongue, extraocular eye muscles, and oral structures as well. And so that could explain why dental work could cause tinnitus in ways that, as the talk goes on, you can understand how this could happen. And so all of these things, these somatic maneuvers, can modulate the tinnitus. They can also induce the tinnitus. So some people who don't have tinnitus, if you ask them to thrust their jaw forward very fast, it produces the sensation of tinnitus. I can do that myself. And we had a human study where we had patients come in, and many people can do it with that particular maneuver. So how could that possibly happen? And so a number of years ago, we started looking at this because of the connection, the somatic tinnitus connection, and the fact that we were studying somatosensory pathways that project to the cochlear nucleus for a completely different reason. And so the person who did this, the initial work, was Jason Zhu, who was a postdoc in the lab. I think, Colleen, you probably remember Jason. So what we did for these studies is we would inject a retrograde tracer. This is a tracer, a protein like biotininated dextroamine is one example, that we inject into the place where we're interested in what projects to this region. So the substance is injected into the cochlear nucleus, which is the first part of the auditory system that gets input from the cochlea. And then what we did was we watched where this substance was transported backwards to the cell bodies of the neurons that project into this region. And what we found, the first set of studies, was that when we looked at the trigeminal ganglion, that there was a bunch of labeled cells in the trigeminal ganglion that actually projected into the cochlear nucleus. Big surprise. No one had ever imagined that such a thing existed. And then we looked up in the brainstem when we saw that there were a lot of labeled cells in the brainstem trigeminal nuclei, and also in the cuneate nuclei, which are the dorsal column somatosensory pathways. So two sets of somatosensory pathways are sending projections directly into the cochlear nucleus. Interesting, because this could explain why people have somatic tinnitus. And so these regions, if you look back to where they get their nerve input from, they get it from the face, basically. And where we see these particular labeled cells, it's predominantly from regions that innervate vocal tracts. So if you record from this region, these cells respond to self-vocalizations, for example. And these cuneate nucleus respond to pressure at the back of the head and neck and upper lip. So there have been some instances of even a stimulation of the median nerve that could modulate somebody's tinnitus. Okay, so we now got very interested in this, and so now we knew that the spinal trigeminal nucleus projected to the cochlear nucleus. So we want to know where these projections go to. So now we put an enterograde tracer into the SP5, spinal trigeminal nucleus is called SP5. And then we watch the stuff that's transported into the synaptic terminals, which are what these little dots are. And so these are cross sections across the cochlear nucleus from rostral to caudal. And you can see that there's terminals sort of all on the edges of this region of the cochlear nucleus. So we can look at that now. We can look at these terminals in a magnified view. And what we see up here is basically we're looking now at the terminals from this region here of the posterior ventral cochlear nucleus, and we can see these large terminals. They're called mossy fibers. And you can also see en passant terminals, so two types of terminals. Now, we were interested to see what neurotransmitter these projections use, because that's going to be important in knowing what the function is of these pathways. So before we did any physiology, we looked at the neurotransmitters, and we found, or Jason found, that they co-labeled with vesicular glutamate transporters. So these were glutamatergic excitatory pathways. And interestingly, that they only co-labeled with a particular vesicular glutamate transporter called vesicular glutamate transporter number 2, so VGLUT2. They didn't label with VGLUT1. And that was interesting because if we put a tracer into the cochlea, and now we wanted to label the projections from the cochlea into the cochlear nucleus, which is what these little green dots are. These are the terminals of auditory nerve fibers. And we tried to label them with VGLUT2, and they didn't label with VGLUT2, but they co-labeled with VGLUT1. So there's almost two completely separate systems that the auditory nerve goes in and labels with VGLUT1. The somatosensory projections label with VGLUT2. So of course now we're going to look at what happens when we deafen the animal. First of all, let's just look at these. So these are VGLUT1 from auditory nerve. And they label here sort of in the magnocellular region of the cochlear nucleus where the auditory nerve fibers end. whereas the V-glut2 labels in the surrounding region that I showed you on the other picture. You can see it over here in red. That's called the granule cell domain. So it innervates mostly granule cells, but also some other cells. And we can plot this distribution of the cells in the cochlear nucleus. So now, as I said, we wanted to see, because now important for the study of tinnitus is, if this projection system is having anything to do with tinnitus, then it's going to have to change after we deafen the animal. And so you might expect that after we deafen the animal, this is going to change in some way that would correlate the changes with tinnitus. So Chang Zheng, who's a technician in the lab, did these studies. And so what we did was we infused kanamycin into the cochlea, and we killed all of the hair cells. And then that led to degeneration of the auditory nerve fibers that go into the cochlear nucleus as well. And this just shows ABR responses at different frequencies. And you can see in the deafened animals, there's no responses. This is just plotting a count of the percent of missing hair cells in the cochlea. So most of them are gone. There's a little bit left here in the apical part of the cochlea. And so what happened, so now this is, you can't really see it very well here, but well enough. So now this is looking at the side, so we deafened only on one side. This is looking at the opposite side, the side that was not deafened, and this is what V-glut1 looks like in the VCM. You can see it surrounds the cell bodies of the major output neurons of this ventral cochlear nucleus. And this is what V-glut2 looks like in those granule cell domains. So what happened after we deafened? Well, all the V-glut1 went away, or most of it. And that makes sense because we killed the hair cells, the auditory nerve fibers degenerated, and so these nerve endings around the cells in the cochlear nucleus went away. But what happened to the somatosensory inputs? Well, they increased. And you can see that over here. So it's sort of like a cross-modal reinnervation. We get lots of glutamatergic input from the auditory nerve, and we get now an infusion of somatosensory, and there's others that come in too that I'm not going to talk about that are also glutamatergic that use this different transporter system. And so we can quantify this, and so what we're plotting here is the difference between the deafened and the normal animal. So if it's above the 0 line, then it means that we got an increase in the V-glut density. And so this is showing the black basically is all the V-glut2, and the green is the V-glut1. So in all these different areas, V-glut1 goes down, V-glut2 goes up. This was at 1 and 2 weeks after deafening, and then we extended this out to 6 weeks, and we basically showed the same thing. So this finding, you get an increase in that change, and it's continued for at least out to 6 weeks. So we wanted to know, okay, we see more V-glut2. Does that mean there's just more V-glut2? Or does that mean there's more synaptic terminals? And not that it necessarily makes a difference in the physiology, but it might. And so what we did was then we put injections into SP5 and the dorsal column nuclei. But we matched injection sizes. So what you're seeing here is a deafened animal and a non-deafened animal. And then we compared the distributions from the deafened animals and the non-deafened animals with same injection sizes in the same places. And this is basically what we found. So this is just showing some of the terminals that we saw in the PVC out and the granule cell domain. And so from the SP5, the spinal trigeminal nucleus, you can see that there was an increase in the number of actual terminals in the granule cell domain, and more specifically in the anterior ventral cochlear nucleus. There was a huge increase in the number of terminals. So not only do we see an increase, but we see a wider distribution of these fibers that don't usually go to this part very much. But now we've got innervation of the central core of the cochlear nucleus, as well as the granule cell domain. And you see a similar thing here with the dorsal column nuclei projections. Okay, so basically, the anatomical side of things say, okay, we're getting this increased innervation from the somatosensory system. It's glutamatergic, it's excitatory. So what does this mean physiologically? And so obviously we wanted to go in and record from cells in the cochlear nucleus while we stimulate the both auditory system and the somatosensory system to see what's happening. And so we do that. This is showing a side view of the brainstem. So here's the cochlear nucleus over here. This is, behind it is the spinal trigeminal nucleus, and the trigeminal nerve is down here. And so we use Michigan probes that some of you will not recognize because you've never seen one like this. So this was a particular probe that two students, a student in mechanical engineering actually teamed up with a postdoc in my lab to design this probe, which is an amazing device. So this is actually our standard probe, this one that just by itself that goes into the dorsal cochlear nucleus. But we also have this combined electrode that can record like 64 channels in the cochlear nucleus, some of them in the AVC and some of them in DCM. So one studies I'm going to talk to you about today are the ones that are just in the dorsal cochlear nucleus. So we've only got 32 channels, instead of 64. And these are Michigan probes, just so you know, Michigan probes, made at the University of Michigan. So this is now, we're zooming into the dorsal cochlear nucleus, and we're zooming into this particular cell here is the principal output neuron of the dorsal cochlear nucleus. It sends its output axons to the inferior colliculus over here. And it receives input from auditory nerve fibers on its basal dendrites, and it receives input from the somatosensory system on its apical dendrites. In addition, there are these cartwheel cells that are inhibitory into neurons that also receive inputs from the spinal trigeminal nucleus. So these where we've shown that these cells project into the cochlear nucleus. So they connect mostly with these granule cells, which the axons of the granule cells are these parallel fibers that innervate the apical dendrites of these two sets of cells, these principal cells and these inhibitory interneurons. And so obviously, I'm going to be talking about inhibitory and excitatory influences, otherwise I wouldn't be going to the trouble to tell you about all of these cell types. Okay, so in our protocol, we put a recording electrode in the region of the fusiform cells in the dorsal cochlear nucleus. And then we put a stimulating electrode in SP5. We've also put stimulating electrodes in the dorsal column nuclei and in the trigeminal nerve itself. But what I'm going to tell you mostly are when we put our stimulating electrodes in SP5, spinal trigeminal nucleus. And when we do that, so you can see here the electrical stimulation artifact, we can either get excitation or we can get inhibition, and sometimes a combined response. So excitation, you can see how that would work. So we would activate granule cells, which would send their parallel fibers to excite the apical dendrites of these fusiform cells, so you could excite the cells and get a response like that. In order to get this inhibition, the parallel fibers would have to first innervate the cartwheel cells, which would then turn the sign to inhibition and provide inhibitory input to these neurons. So that's how we could inhibit the cells. They would have to go through cartwheel cells. And in fact, when we do record, you can't really see it, it's not really lined up very well here, but the latencies of the inhibition are 1 to 2 milliseconds longer than the latencies of the excitation, which makes sense that there's an inhibitory neuron in between them. Okay, so now we're not only interested in the responses that we get from stimulation of the somatosensory system. We're interested in the responses to sound, and more specifically, we're interested in what happens when you combine the stimulation. And so in order to do that, here's our circuit again. We're stimulating this, just like I described. But now what we're going to do is we're going to pair the somatosensory stimulation with the sound. So in this first example I'll show you, we basically just put the trigeminal stimulation first, and then we follow that with sound. But remember that when we're stimulating here, that these neurons are pretty far away from these, so it's going to take some time for them to actually be activated. So putting this, before this, could mean that they're going to be activated at the same time. So this is one example of what happens. This is a post-stimulus time histogram of a response from a dorsal cochlear nucleus neuron to a noise burst. It's just going psh, 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 and we're building up the responses to this. Now when we precede that with the trigeminal stimulus, you can see that the firing rate is decreased substantially. And when we move the trigeminal stimulus closer in time to the auditory stimulus, we get an even smaller response. So this tells us that in this particular neuron, we're inhibiting the neuron with trigeminal stimulation. It also tells us that this inhibition is interstimulus interval specific. So it depends how far away the stimuli are from each other. And in this case, we're actually stimulating the trigeminal nerve, trigeminal ganglion. So it's the same thing when we stimulate the trigeminal nucleus. Okay, so we're going to come back to this combined auditory somatosensory stimulation. And so this sort of just forms the backdrop. Yes, we can activate these cells, so sometimes we can actually increase the firing rate when we pair them. So now we want to sort of jump forward and see what happens to the system when we deafen the animals with the intent of them having tinnitus. So the first studies, we just deafened the animals. We didn't measure whether they had tinnitus or not. And so in this first study, which we did quite a few years ago now, we used a very loud sound, and we produced a permanent threshold shift in these animals. So a lot of these cells didn't respond much to sound anymore, but we could measure their spontaneous rates. And so here are control spontaneous rates. Here are the deafened animals 1 week after the deafening, and you can see their spontaneous rates increased. And in 2 weeks after, they were still increased. And so this increase in spontaneous rate had been used by several groups, now including ourselves, as a physiological correlate of tinnitus. So when there's no sound stimulation, the neurons are firing at a higher firing rate, and this is generally only those neurons with best frequencies within the noise damage region. And so it's a frequency-specific increase in spontaneous rate that could signal tinnitus to higher auditory neurons. So it's one of the physiological correlates, and it since has been correlated with behavioral evidence of tinnitus in several different groups. So we found this increase, so this was nothing new, we just replicated what these groups had found. But what was new is what we did next. And so because these neurons, this was a heavily deafened preparation, unlike the future ones I'm going to tell you about. So we couldn't say, we wanted to know which cell types were responsible for these increases in spontaneous rate. We couldn't measure responses to sound, but we could measure responses to somatosensory stimulation. And so that's what we did. And so this is the control group. And so when we stimulated the trigeminal ganglion in this case, we either got excitation or inhibition, like I showed you, and also combined excitation inhibition. Or we got no response, and that's what we're showing here. So the spontaneous rates of all of these different neurons that responded differently are all about the same. So if they didn't respond to somatosensory stimulation, they had maybe a slightly lower firing rate, but none of these differences were significant. Okay, so now we expose the animals, and we look after 1 week, and it sort of looks like these ones are coming up here. So these are the ones that had either excitation, inhibition, or excitation. So some kind of excitation from the somatosensory system. But at 2 weeks, it was pretty clear that it was only the ones that had excitation, inhibition, or excitation alone that were producing the increase in spontaneous rates. So this was for us a very important finding, because it basically said that the neurons that increased their spontaneous rates were those that were excited by the somatosensory system. And so obviously we wanted to look at this in more detail. We also showed actually that these neurons, after noise damage, the neurons had lower thresholds in all 3 of these groups, the excitatory, combined, and inhibitory. The thresholds were lower. So this actually goes together very well with the anatomical studies showing that there's more inputs coming in to the cochlear nucleus, more excitatory inputs coming in. So there's an increased excitatory drive, plus now you're actually making these neurons more sensitive, which relates also to the spontaneous rates, that we may be lowering their membrane potential so that they're more responsive to the somatosensory stimulation. So basically, just to sort of catch you up so far, what I've showed you is that somatosensory stimulation can suppress or enhance the firing rate. It can also change the spike timing, which I don't have time to show you. and of sound-driven responses in the DCN. So in addition to just the plain responses that you get from stimulating the system, this is integration. So it changes the actual firing rate elicited by sound. So these are potential mechanisms for altering the loudness or pitch of tinnitus, if we can change the evoked responses. It also results in elevated spontaneous rates in somatosensory-driven neurons and lower thresholds to somatosensory stimulation. So one contributing factor is the increased presence of excitatory terminals that are signified by the VGLU2 findings that I showed you. So now we're obviously now interested to know more specifically. What I've shown you so far is really only the effects on noise damage. But now we're interested in how this plays a role in tinnitus. And so a postdoc in the lab, Susanna Damo, spent 4 years in the lab doing this very extensive study. So what she did was, as opposed to what we'd done before, is we blasted the animals and made them deaf. In this case, we wanted to give them a temporary threshold shift. So we wanted the thresholds to be the same in our experimental group and in our control group. And so we developed a noise damage procedure where we used a quarter octave band that was centered at 7 kilohertz, 97 decibels for 2 hours, just in 1 year. And this is just showing where that noise band falls in the guinea pig audiogram. And so this is what, now we're looking at the threshold, ABR threshold shift. And so the left here shows, the orange here shows the threshold shift a couple of days after exposure. And then this is 1 week after exposure, everything has come back to normal in terms of threshold. Now in terms of suprathreshold function, along this whole study we did ABRs that I don't have time to show you today. But that's going to turn out to be an interesting study on its own because it shows some of these suprathreshold changes. Not exactly as what Charlie Lieberman's group showed. But anyway, the right ear didn't show any threshold shifts, and if anything, the thresholds got better on the right ear, which is also a topic of another grant. So we got frequency-specific temporary threshold shifts. Thresholds come back to normal very soon, like after less than a week, the thresholds are back to normal. And at the same time, we used the gap detection-induced reduction of the acoustic startle as a behavioral test for tinnitus. This was developed by Jeremy Turner in rats, and we've developed it in our lab, which we think is a very effective test for tinnitus, especially in our model of temporary threshold shift. We've spent a lot of time modifying the parameters of the gap detection procedure so that we keep our startle amplitudes very constant across the whole study. And that's easy to do when you do a temporary threshold shift as opposed to a permanent threshold shift. So this test goes like this. It's basically you have a background noise band, and then you present a loud startle stimulus that makes the guinea pig jump. And we measure this with a startle plate. So this is a rat, I think. Is that a rat or a rat? It's a rat, okay. Now if you insert a gap within this noise band, it's sort of like it warns the guinea pig or the rat or the rabbit that it's going to get a startle stimulus, and the startle is reduced in amplitude. So the theory is that Jeremy developed is that if the animal has tinnitus and the background noise is at the right frequency of the tinnitus, that it's going to fill the gap and that the startle amplitude is going to go back up to normal. And so that's what we base our test on. We've done a lot of tests, and we've published 3 papers on it now, so we think that in our hands this is a reliable technique for testing tinnitus. And so these are some of the ABR threshold shift results and tinnitus results before I get into what happened with the somatosensory system. So what you're seeing here is this is the spectrum in gray. This is the spectrum of the noise that we used. Then this black curve here is the ABR shift in the left ear. And this is the ABR recovery in the left ear, and the ABR that you've seen already, this reduced threshold, in the right ear. This is what she saw with spontaneous rates. So it's sort of, it overlaps actually very nicely the spectrum of the noise. It's a little bit slightly lower than the ABR shifts. So this is the spontaneous rate spectrum, the increased spontaneous rate that we saw, specific to the neurons with best frequencies close to the noise damage frequencies. Okay, so now after we did all of this, now at the end of 3 months now, where we've basically checked ABRs all along the way, checked gaps all along the way, now divided our animals into those that have tinnitus and those that don't. So about half of the animals got tinnitus using this technique, and the tinnitus frequencies were basically in these regions, here, 8, 10, 12, 14, 16, but mostly at 8. They're mostly occurred at 8. And so now we went in and put our multi-channel electrodes in the dorsal cochlear nucleus, a stimulating electrode in the spinal trigeminal nucleus. So first just looking at the auditory responses. The thresholds of the single units and multi-units came back to normal. This is showing spontaneous rates of those individual units. So close to the noise band, slightly, close to and slightly below had elevated spontaneous rates. And in addition, an interesting finding was that when she plotted the rate level functions of individual neurons within the cochlear nucleus, the black shows the normal animals. So now we're just comparing noise damage to non-noise damage. You can see that the rate level functions are much steeper. And this is sort of like, I know there's audiologists in the audience, this is sort of like hyperacusis. So this may be one basis for hyperacusis that is often associated together with tinnitus. It may or may not be the same mechanism, but you often see them together. So this is just showing, again, this is now mean data. So we're looking at rate level functions. These are now dividing them up into those that displayed tinnitus according to the method I showed you. So the ones that have normal and the no tinnitus group showed this rate level function. The ones that had tinnitus showed this elevated, slightly steepened, and elevated, including spontaneous rate function in fusiform cell responses. This is just showing a post-stimulus time histogram demonstrating this enhanced responsiveness of these cells. Okay, so now we know we've repeated that these neurons show increased spontaneous rate. They show hyperacusis type responses to sound stimulation. But what happens with multisensory integration? And so what Susanna did in this study was we had already shown that when you stimulate the trigeminal system, you can either decrease or increase the firing rate in response to sound. And that's what is shown here. So we presented a sound first, and then we presented a bimodal stimulus, and we recorded the sound in response to that. And so in this case, you can see that it's suppressing, but sometimes it's enhancing. And then what was an addition in the study was we added long-term effects. So after 40 minutes, now we recorded again from the same neuron or group of neurons, and showed that we still maintained this depression, and in fact it was even more depressed, or enhanced. So we could either enhance or depress. So we now call this a long-term effect of somatosensory stimulation. We only tested out to 60 minutes at the most, so we don't know how long. If you stop doing the stimulation, you can eventually recover it after about 2 hours. If we just stop everything and then keep recording from that neuron. So it's a long-term effect. And so we're calling now this somatosensory plasticity, or multisensory plasticity. And then what came out from this study, basically, is if this was the immediate effect that's plotted here on the left, and this was the long-term effect. And so now we're dividing it up into a control group. And so we've sort of scaled this down here, but there's a 0 line there, and this shows that we can enhance the firing rate in some units, and we can depress the firing rate in other units. And this replicates what we showed in our other studies. It's about half that are enhanced and half are suppressed. And this happens at all levels. So now we're looking at all different levels of sound. So now, even in the controls, if we wait a while, we see an enhanced effect. And so we see some enhancement and quite a lot of suppression, especially at the higher levels. Now, let's look at the ones that didn't have tinnitus. So in the ones that didn't have tinnitus, we're still seeing an inhibition that looks, so this is not just inhibition, this is somatosensory-driven inhibition, or bimodal-driven inhibition, or suppression. But we're seeing some enhancement here at sort of mid-levels. So there's more enhancement in the no tinnitus and about the same amount of inhibition that you see in the normals. But if you look at the tinnitus group, you can see enhancement all the way across all levels. And the inhibition is just about gone. So this fits with previous studies, is that, remember I told you that in order for the somatosensory system to inhibit those cells, you have to have an inhibitory neuron in between. And other studies have shown a decrease in glycine in the cochlear nucleus, and those cells are glycinergic. So that sort of fits with the cortical cells playing less role, and now we're getting more direct innervation from the somatosensory system directly to those fusiform cells. But it's enhanced in the animals with tinnitus across all levels. So when she quantified this, if we just look at the long-term effect, and we compare, here are the control animals, so we're adding up all instances of excitation and inhibition across level. So the controls are definitely dominated by suppression from the somatosensory system. The no tinnitus animals have about half and half, but the tinnitus have mostly excitation and very little inhibition. So that was the main outcome of this particular study, was that in the animals with tinnitus, they're showing increased excitatory drive from the somatosensory system, or from bimodal integration. Because you couldn't just do this with somatosensory stimulation alone. I don't have that data to show you here, but when we stimulated the somatosensory system alone, we didn't see all that much change, and when we stimulated repeatedly with sound alone, we also didn't. So it has to be combined, somatosensory auditory stimulation. So far, just to recapitulate, in this study and in the previous one, auditory nerve innervation is decreased. So even though our temporary thresholds, the thresholds have come back to normal, we're still assuming that, like Charlie Lieberman has shown, that there's some damage to synaptic ribbons, and so there's going to still be a reduced auditory nerve input, which is going to trigger the enhanced somatosensory stimulation. So we haven't measured that. We haven't measured VGLE1 and VGLE2 with a temporary threshold model shift. But we have measured physiological responses that I've shown. So with the long-term, the high exposure situation and permanent threshold shift, we get increased spontaneous rates in those neurons receiving somatosensory input. So with the narrowband and TTS, we see a CF-specific increase in spontaneous rate, steeper sound-driven rate level functions, and long-term enhancement by somatosensory stimulation in the fusiform cells. So now we are interested in, now this is the long-term effect, so it's plasticity. Well, what is it? So now the next set of studies wanted to investigate what it is. So now we're back to our circuitry in the dorsal cochlear nucleus. And the findings that we've seen now, this long-term effect, they seem to have about the same time span as what Thanos Xenopoulos demonstrated in the slice preparation, which is this. And he showed spike timing dependent plasticity in fusiform cells in a slice preparation. And what that is, what spike timing dependent plasticity means, is that if you repeatedly pair a spike, so they activate the cell, and then they depolarize the cell, either one before or after each other. So in this case, the spike is coming before the synaptic activity. So it's like they're activating this cell, making it spike, and then they're depolarizing these apical dendrites. And so when they precede the synaptic activity with a spike, they get long-term depression. This lasts for about a half an hour. But if they reverse the order of the synaptic and the spike activity, they get long-term potentiation. And that's what defines this as spike timing dependent plasticity. So then we wondered, okay, we've been just using basically one value. We've been preceding trigeminal stimulation, which is equivalent to the presynaptic activity, or the synaptic activity. And the sound activation is like what they're doing with the spike. So it's really the same thing that we're doing and what they're doing, except we're doing it in vivo. And so we decided to investigate that by changing the order and timing and intervals between the stimuli. So our hypothesis was that spike timing dependent plasticity is necessary, and maybe the mechanism underlying bimodal plasticity that I've shown you. And so, to do that, we had SP5 before tone, and then we gradually increased the, we reversed the order and increased the distance between the tone and the SP5. So we're basically replicating this protocol that Thanos used in his slides. And so this is what the paradigm looked like. We presented the tones, then we followed them with a bimodal pairing protocol, and then we looked at the tone response again. And the bimodal pairing protocol was all of these different things. We either have SP5 before tone onset or tone onset before SP5. And these do overlap, you know, but that was how we did the protocol. So we're looking at onset, stimulus onset. So this is what responses look like when we do this. And so let's just look at this one over here. So this one is showing, so the blue is showing basically the response to the tone alone before, and the red is showing the response to the tone alone after the bimodal pairing protocol. So you can see here that in this case it caused depression. But if we change the order and interval, for example over here, now SP5 is coming before the tone in this interval, we're seeing enhancement. So that's what it looks like. Maybe it is spike timing dependent plasticity. So what we did was we calculated the average firing rate for each of these conditions, and then we plotted them as a function of the interval here. And you can see that basically some units we can exactly replicate the spike timing dependency that you see in the slides. And of course in vivo it's not quite that simple. So, and this is the work of Seth Kohler, by the way, who just graduated and went to Hopkins. And so it's hard to get him to finish up the next paper, but we will. And so what Seth showed was that there basically were 4 different kinds of responses that we could get. We could get one that looks very similar to this one. That's this one here. But we could get the opposite as well. So we could get Hebbian and anti-Hebbian. We could get ones that showed mostly enhancement and ones that showed mostly suppression. These are multi-unit responses because it was easier to hold the multi-units over the length of period that we needed for all these intervals, which were randomized. And these are some of the single-unit responses in these different groups. So it was really a tour de force to hold a unit for that long, that we had all of these conditions, you know, for, 5 units, 7 units here, and only 2 in these groups. So very big work on Seth's part. So what we can conclude from this is that spike timing dependent plasticity is one mechanism underlying the long-term somatosensory enhancement that we see in the tinnitus animals. In the normal system, what I've showed you, we get anti-Hebbian and Hebbian timing rules, both of those different rules. In noise-damaged animals with tinnitus that I'm not showing you yet because we haven't published them yet, but these rules change, and we get predominantly Hebbian rules. And so this now gives us a very nice tool for doing some sort of intervention with tinnitus, because if we can modify the firing rates of specific groups of neurons, we can build a device, which we've actually already built. And it's a patent application, so I'm allowed to talk to the camera about this. And so the idea of this device is that basically we would replicate what I just showed you, but not with the rules that I just showed you, because the animals with tinnitus have different timing rules. So the device would be based on the timing rules in the animals with tinnitus. And so what we're going to do now is we're going to be doing bimodal stimulation with a device, and we've got some groups of guinea pigs. We'll induce tinnitus in some of them, and in some of them we won't, and we'll test these different bimodal pairing intervals to see whether we can reduce the amount of tinnitus and the physiological correlates of the tinnitus as well. So that's an ongoing study that hopefully we'll have some results on that in about a year's time. And then the idea is to take that to as a human device, like an iPhone app, that the person could tweak these. We'd have a sort of set parameters. The person could tweak these so that the auditory stimulus matched their tinnitus, and the intervals could be tweaked until it makes their tinnitus reduce. And then they could use that for a couple hours, you know, a day. And anyway, you know, it's just you know, an exciting adventure to see what, you know, if theory actually is going to fit with practice. So it's time to stop. And these are the people who did the actual work. So these are people in my lab. The ones in bold are the ones whose work I actually presented to you. So it's like I said, Seth Kohler has now left the lab, but I'll be presenting his work for quite a while, so he's going to stay up here for quite a while. And these are our funding sources, and these are our various different collaborators.
We have time for questions, comments.
So when we looked at patients who had tinnitus in Buffalo, we found that nearly all of them could modulate their tinnitus with oral facial movements, particularly the jaw. Unfortunately, almost all of them could change either the pitch or the intensity of the tinnitus, but it was almost none could actually decrease the tinnitus.
Right, well, I wouldn't expect them to be able to do that because that fits very well with what I just said to you about somatosensory stimulation alone. It doesn't do anything. And so, so that's why like 10 years of work may pay off in that people have tried somatosensory stimulation. It doesn't work because it won't work because you can't induce plasticity in this particular system, in the cochlear nucleus, in this particular system, unless you combine the stimuli in a particular way. And that's what's inducing the plasticity. What I haven't showed you is that the study that I showed you of Steph's, we have identical data from auditory cortex that we reported simultaneously with this. In both the normal animals, what I showed you, and in the deaf and plus tinnitus animals, that will be, you know, that will be in my next talk. But the cortex data also shows spike timing dependent plasticity with this bimodal protocol. We haven't yet seen what happens to the timing rules in the cortex with the deafened versus the normal and the deafened tinnitus versus the deafened non-tinnitus. So I think that'll be more data to feed into our potential device because it could be that the cortical neurons do something slightly different. And if that's the case, who do we believe, DCM or cortex? You know, maybe we have to try both. Because they're not identical. And the cortex responses actually are much more homogeneous because there's less cell types. You know, in the DCM, all of these different rules that you see are based on the different cell types. You know, and there's deaf cerebral cell, even fusiform cells. Some have less inhibition, some have more inhibition. And we correlated the particular timing rule with the amount of inhibition that neuron gets, looking at their tuning curve. So, you know, there's a huge amount of work still that one could do that, you know, that other labs should do. And, you know, I think only by doing it that way are we going to actually, you know, be able to help patients.
Can we get one more?
Yeah.
Okay.
Yeah,
So
two more. Else
on that same vein where you're talking about ACCN, one of the interesting things, I was very happy to see that your actual controls were animals that had been noise exposed as opposed to just comparing noise exposed plus evidence of tinnitus versus normal. So that was...
Yeah, well, it's a very important question. Why do some people get tinnitus and others don't for the same hearing loss?
So my question was, did you also look at, if you're using the sort of the Lieberman style noise exposure, did you look at super threshold measures in the ones that had tinnitus versus the ones that didn't, to see whether or not they differed in their super threshold measures, and that might explain some of the differences?
Yeah, did you come to my poster in Aero?
I walked by your poster.
So like I said, like with Susanna's study, you know,
Yes.
we recorded the ABRs continuously over that time. We did actually publish that data, but she didn't have big enough data sets to do what we really wanted to do. So with Seth's study, we did input-output functions right after the exposure, 1 week after, 2 weeks, 4 weeks, 3 months. So we have ABRs all along the way. And we should have a board. We do have a board here. So I'll show you what, I mean, it's really quite interesting what happened. So Maybe not. I'll tell you that. So the rate, I'll draw it here in the air. Okay. So if we look at 3 months afterwards, which is sort of equivalent to when Charlie would be measuring, or when the human studies would be measuring, then we get an input-output function like this, well, like this for the normal animals, and it's sort of like this for both the tinnitus animals and the no tinnitus animals. So we can't really tell the difference out here. But when we go back in time, and so now if we look at 2 weeks after the exposure, then, so the normals are like this, the ones that don't have tinnitus are like this, and the ones that do have tinnitus are like this. So there's something going on early, some increased excitation or whatever it is. My guess is it's lateral efference or something like that. Because clearly, I mean, all of these, you know, in the end they're like this. So either they degrade slower, or there's like more or less efferent input into the tinnitus of the non-tinnitus group, but something's going on earlier that then brings it back to, at the time when other people measure it, this is happening. So that's wave 1. So wave 1, there may be some predictor of what's happening here, but actually I think that may be more related to hyperacusis, because that's at all frequencies, not just within the noise band. So now if we go to wave 2, which is, and by the way, we did this in both ears, which nobody else does either. So this effect and this effect, it happens in both ears, not just one ear. And wave 2, which is really interesting. So wave 2 at this time point, it shows a frequency-specific elevation in the tinnitus animals only, in the noise band of the TTS. And then if we go, and this is just in the left ear, the right ear is just normal. Now we go back to 3 months, and this thing, you know, in the left ear has gone away, but now it's switched to the right ear. So there's a frequency-specific increase in the amplitude of wave 2 in the opposite ear. So that's just very interesting to us because of the contralateral excitation. And, you know, I pointed out to you that the ABR thresholds are actually better in the contralateral side. So I think that there's, other than, you know, in addition to the somatosensory stuff, of course, it's not just about somatosensory. Everyone thinks that I only know, you know, like everything's somatosensory, but this, you know, cross-pathway changes are, you know, are very interesting. So what I'm talking about is that there's a pathway between the cochlear nuclei that's usually inhibitory. And when we deafen one side, this pathway becomes excitatory. So it's all predicated on unilateral hearing losses. And there's a total imbalance in the system, and I think that's what is active here in, you know, where wave 2 is the one that's showing up these increases in tinnitus, and then it switches to the other side, you know, at the end. So really the only thing that's predicting tinnitus in the end is wave 2. Because wave 1 is like this for both the tinnitus and the non-tinnitus animals. Although over here, it's only in the tinnitus animals. But it's all frequencies. So I think that this is predicting hyperacusis. And this CN wave 2 is predicting tinnitus. Long answer to your question.
I want to continue hijacking, but I want to the chance to.
Any other questions? Yeah.
I have a more general question. If I understood everything correctly, the idea is when you do this noise induced damage, you're causing tinnitus, right? Or tinnitus because of the elevated activity in that pathway. So it's not like whatever is happening in the cochlear nucleus is causing tinnitus. It's something else. So what you're really looking at is how there's a somatosensory input that's modulating that. You're relating it back to these human studies, which when you pop your jaw and all this other stuff, you can get that to happen. That's interesting, but that seems like the least interesting aspect of all of the data you presented, because I don't believe these connections from the somatosensory system are there simply just to modulate tinnitus when somebody actually expresses it. So it must have some other function in the animal to be there in the first place.
Right, it does, but
And
that's
so
just not what I talked about.
Right. So is it related to why when we speak versus when we hear our own voice, we hear different differences? Is it through this pathway?
You're a very clever man.
I mean, that seems to me like
Yeah,
that's a very interesting question to be looking at. Not that tinnitus isn't important.
Yeah, that is one of the proposed functions, is that these non-auditory inputs, including somatosensory, act as an adaptive filter. And adaptive in the sense that the filter is going to be different depending on whether the sound is coming from outside of you or within you. And so in the situation where you're producing your own self-vocalizations, that's by definition is a combined stimulus to the dorsal cochlear nucleus, because you're getting the vocal input, somatosensory, at the same time as you're hearing your own voice. And so that's likely going to suppress the activity of those particular neurons that are responding, so that you're not overwhelmed by the loudness of your own voice, so that you can attend to things outside. And that's actually very similar to the function of this, a very similar kind of circuit in the electrosensory lobe, which is very similar to, this is like a cerebellar-like circuit. So the circuitry is very similar, the actions are very similar, and we haven't actually proved that yet, but it's one of the things that we want to do is the following experiment where we make the animal vocalize, because self-vocalization is really different. And we can make the animal self-vocalize by stimulating certain parts of the brain, like a region called the periaqueductal brain, that will make the animals vocalize, and then we can record that and play that back to the animal, and look at the differences with the exact same sound, and the differences in the responses in the DCM. But, we're funded to do this. There's lots else that we do that I don't talk about here that isn't necessarily have anything to do with the somatosensory system. But yeah, that's one of the functions. Another one of the functions that comes from the work in Eric Young's lab, where they used the cat. And in fact, they were the first people to look at the effect of stimulating the somatosensory system on the auditory system. And the cat was, a very specific model because it uses its pinna to help localize sounds. And, not many other people, like species like people, do that. And guinea pigs don't do that, and there's not many dogs sort of do it, but cats do it best. And so what they showed was that that the activity in the DCN of the cat was very linked to the pinna movement. And then they got the biggest responses in their preparation when they stimulated regions that were activated. So in fact, they defined their regions that they stimulated based on whether they were activated by pinna movement. So one of the things that comes out of that lab's work is that it's important to know the location of your pinna if you're a cat, so that you can orient to a sound, or it's giving you information about where the sound is coming from. So in species like rats and humans, not necessarily that rats and humans are the same, but they move their necks. And so that would be more like the dorsal column input, would be more for the sound localization. The trigeminal input that seems to be, in our hands, more focused on the vocal structures, it's probably what you just suggested, is for the suppression of self-vocalizations. There are also inputs from pontine reticular formation and the pontine nuclei. They also send inputs into the cochlear nucleus that also be Bluetooth positive. And so that could be, cardiac, like suppression of heart rate and respiration. You don't want to hear yourself breathe. You don't want to hear your heart rate. So yeah, that's exactly what it's for.
Last question.
Yeah. In the clinical, usually the patients with tinnitus, they have a hearing problem. Can we use your model to give like right explanation, the mechanism, why they both connect to each other so close?
You can if you want. I'll send you the papers. I mean, it is. This is one of the mechanisms. It's not. It's like in the tinnitus world, you'll hear one story depending on what that lab is working on. It's a very complex disorder. And so we've spent many years working out this particular system. But, we're also interested in the input from the other side, the cochlear nucleus. The efferent system, I'm pretty sure, is playing a role in tinnitus as well. And then there's, you know, if you move up in the system, there's other aspects to tinnitus, like, you know, some people have tinnitus that bothers them. The same loudness of tinnitus, one person, they will jump off a bridge, and the other person could care less. And so why is that? And that's recently become a big and popular topic of investigation as well. So those people, those groups are investigating limbic system involvement in tinnitus. You know, maybe a little bit harder to investigate. The cochlear nucleus is this great circuit. We know that we can put an electrode in there, and we know by the response which cell we're recording from. And that's why I love the cochlear nucleus, just because it's, you know, the circuitry is so well known, and I just like working there. But that doesn't mean that we don't. We've also put electrodes in the inferior colliculus and in the auditory cortex, and now I have somebody in my lab whose focus is on the auditory cortex. So, you know, another focus of groups is looking at interactions between the thalamus and the cortex, and you know that maybe there's a lack of there's too much synchrony between those two circuits, and so that's another place to look. But it could be that it all starts here in the cochlear nucleus, and so you know if you've got spike timing dependent plasticity that is shaping the responses of these output neurons that go to everywhere else, it has to influence them. Not that there isn't processing, you know, that goes on there. So it's like each time you go up, you're building a layer of complexity. But if you don't understand this, you can't really ever understand this, is my view of it. Now, if you talk to a person who just records there, they say this isn't important. You know, so it depends, you know, you can go to read the literature and form your own opinion. It's my opinion that you have to look at the source of things. You know, and then move on and try to integrate, you know, how this influences this, and what is conserved and what is added. And that's sort of how we're looking at the auditory cortex stuff. But ultimately, you have to look at how these all influence behavior. You know, and so there's, you know, it's pretty clear that in tinnitus there's a decreased inhibition, both from glycine and GABA, depending on which level you're at. But it's pretty clear also from our studies and from some other groups that there's an increase in excitation. And so if you want to do, you know, like develop drug treatments for tinnitus, which I don't think is a good idea anyway because you're affecting everything, but if you were, you'd have to target glycine and GABA and NMDA receptors. You know, and the trouble with all of that is that you're doing that and the whole brain is affected. So if you can have a treatment that specifically activates those cells that are involved in the disorder, that's better. It's just hard to do. But maybe you can do it with a device better than, you know, I mean, ideally, what you want to do is you want to prevent the cochlear damage. Or if you can't do that, you want to regenerate the auditory nerve fibers so that you're maintaining the input into the cochlear nucleus. And then you combine that with a device, you know, and maybe you've got a good treatment.
Okay, I think we're going to stop the group questions and continue in private because I think there are more questions.